Okay. Anyway, it's a pleasure to welcome Adam MacLean, who's going to give us a different perspective from what we've been hearing. This is non-genetic paths of tumor escape. So I fear plasticity is on the cards.
Thank you very much. Hi, everybody. Thank you to the organisers, to Sandy and Kristen. I'm very excited to be here and tell you a bit about work from our lab on non-genetic paths from tumor escape. So that's it. That's my talk. I'm only kidding. Despite thinking this daily, of course, I've cut off the quote from Dobzhansky that nothing in biology makes sense except in the light of evolution. Many of us have remixed and reused this many times over the years, and I think with good reason, because it's really important to keep in mind almost all the time in the research that we're doing in biology, and of course, equally true in cancer, that nothing in cancer makes sense except in the light of evolution. And indeed, the most exciting research directions today, and some of the focus of this conference is on evolutionary therapies and adaptive therapy. But this is a session on cell state plasticity. And so I'd like to ask you to join me in flipping Dobzhansky for a minute to consider the question, what might we miss if we don't look beyond cancer evolution and consider other sources of heterogeneity within a tumor? So I'm going to introduce these sources of tumor heterogeneity across scales. And why is it important? So I guess I'm going for more of an educational than a moral tale when it comes to my talk. But if there is a moral, it's this. It's like wherever there is heterogeneity, the cancer will exploit that for its own good and co-opt these pathways for its success at the expense of the host, of the patient, of us. So these sources of heterogeneity, we begin with the genetic variation in the tumor gives rise in the context of oncogenic mutations to cancers. I'm showing you here epigenetic heterogeneity. This is an example of like the promoter of one gene, its chromatin accessibility. You can think of other epigenetic marks like methylation and how these change over processes such as stem cell differentiation. Then moving up the scales, we get to transcription, which defines the gene expression, and you can look in single cells across tissues at the many different genes that individual cells express. At the cellular scale, you can look inside of a tumor and see that it's made of not just cancer cells, but a whole myriad of immune cells, fibroblasts, and all sorts of other cell types in there too. And counting the cells is step one, but of course they don't just sit there, they talk to each other, and cell-cell interactions within the tumor defines an essential source of heterogeneity. And finally, of course, there's the microenvironment, and the larger environment and the tissue or organ in which a tumor is situated. So I'm going to give you a couple of examples of recent projects in my lab. We spend quite a lot of time at this kind of high scale of cell-cell interactions and spatial location environment, and then zoom in on the transcriptional scale. So sites of metastasis can completely dictate responses. I'm showing you some results from our clinical collaborators who have a phase 1b expansion cohort treated with immunotherapy, anti-CTLA-4 and PD-1 combined with this epigenetic drug entinostat. This is metastatic breast cancer patients for whom the site of metastasis, as you can see, is like dramatically affects what happens to the tumor. So if you get a met to the lung or the lymph node, your outcomes are really pretty good. But if it metastasizes to the liver, that's the first one there shown in purple, you have much worse outcomes overall. So a very simple, high-level environment dictates a response to a large extent in this case. Environment can also determine if a given treatment is going to be successful. Here I'm showing you from the same cohort, a single patient. We're looking with spatial proteomics at the cellular locations of subtypes of immune cells within the tumor. The green is CD8 T cells, the pink is the macrophages. And when we quantified how these distances change across all the patients before and after treatment, we saw something really striking, which was that the co-occurrence probabilities go down significantly only in responders. Okay, so co-occurrence is like 1 over the distance. It's the likelihood of these cells to be actively next to each other and interacting. And so this is exactly what we want to see when the macrophages are interacting less with CD8 T cells. And this is the release of immune suppression that allows the effector CD8 T cells to become cytotoxic and start killing cancer. So these are just a couple of different ways that environment it can dictate response. But now I'd like to zoom in on cell state and cell state transitions within a tumor. So cell states are both discrete and continuous. So what does that mean? That doesn't seem to make much sense. I'm showing you here at the bottom a couple of microscopy images of 2 different cell states. On the left are epithelial cells. They really like each other. They stick together in clumps, and they like to grow. On the right are mesenchymal cells, which are loners. They mostly exist by themselves, and they're often motile and migratory. And the epithelial to mesenchymal transition, or EMT, is a really essential process in development and in cancer because it primarily drives metastasis. Okay, so from a cell state perspective, then this seems easy. We've got 2 states. They're discrete. They look completely different. The cells are different functionally. Everything is fine. No worries. Well, so there's not 2 states. Here is one other state. This is an intermediate EMT hybrid state in which you see cells that look kind of epithelial and kind of mesenchymal. Okay, but they're quite distinct. And this is not just transient. Okay, you can kind of watch these cells and they can be stable and they can persist in this intermediate state. Okay, so maybe we just have 3 discrete states. Can we stop there? Maybe, maybe not. If we look at transcriptional data, we're now talking about quantifying the gene expression in lots of single cells as they undergo EMT, you'll really see something that looks like a continuum without a discrete separation. And some people argue that actually discrete picture of cell states is not a correct way to view this system. Furthermore, we don't just have one trajectory, but we have multiple going through this process of EMT, and they correspond to kind of different periods of stability or different sizes of the relative states, the E and the M and the intermediate. And we're really going to be interested in these hybrid or intermediate EMT cells. And the reason is, in a canonical picture of metastasis, cells undergo EMT, and the mesenchymal migratory ones are the ones that are able to seed new metastases. But actually, there's a really growing body of literature that suggests that the intermediate cells, the hybrid cells, which are migratory yet retain an ability to stick together to a certain extent, as the epithelial cells do, and to proliferate more, are really the ones that are the highest in terms of their metastatic capability. So kind of like identifying these, finding biomarkers, and potentially kind of blocking these states could be a really powerful way to reduce or stop metastasis. So we first looked at this just by quantifying EMT states across a lot of different, mostly cancer cell lines, a couple in vivo data sets, just using a global gene expression EMT score. And just show you a couple of things about this distribution. It gives you kind of the start state and the end state, and you see these are different. So the epithelial and mesenchymal states themselves are not the same, and also the width or the kind of extent of this distribution is also changing dramatically between samples, although there are some kind of reproducible things, like when you stimulate with the same growth factor, you get a similar response. We use this kind of compendium of single cell data sets describing EMT to fit a really simple mathematical model with just 2 parameters, the E to intermediate state transition and the intermediate to M transition. And we see this kind of like this inverse proportional relationship, which makes sense. And we use those relative parameters to try and look for biomarkers of the highly metastatic intermediate state cells. So we're kind of looking at Spearman correlation here for a couple of these top genes, stratophin and NRG1, which are really highly correlated with the parameters of like transition, suggesting that these are biomarkers of the dynamic changes between states, which again, we think is like along the continuum of the cell states, likely to mark for some of the most metastatic cells and the ones that are kind of also hardest to identify in samples. So this is a kind of way in which we can just get biomarkers, but we're still not able to, from this like simple gene expression data, alone able to really characterize the networks that are controlling a cell state transition. Okay, so to go back to basics, how is a cell state transition controlled? Well, the state or fate of a cell is controlled by the genes it expresses, i.e. its transcriptional state. And this gene expression, in turn, is controlled by transcription factors via gene regulatory networks. You can think about these genes that encode for proteins that bind to the DNA and activate or inhibit the expression of other genes. Okay, so we take a cell, let's say we take an epithelial cell, we look inside it, and this is what, you will not see, but as a modeler, this is what you will see. Okay, you will see a network of transcription factors and targets that turn on and off genes in a way that permit the cell to express different things and change its state. Of course, these networks don't stay small. This is a kind of snapshot of a snapshot, and they get really big really quickly. This is still a snapshot of a network describing EMT development, and I think this is really scary. And going towards some of the themes, this is certainly not small. You can imagine how many parameters it would necessarily have. But don't worry, there's not 100 parameters in this talk. Cell state transitions can be described accurately by small GRNs. And this is an example for EMT. This is a 4-state network of interacting transcription factors and microRNAs. What do I mean by small? I mean, this is, of course, kind of hand-wavy, but we're talking about like less than 5 nodes or species in a network. And the reason why, mathematically, is just that the systems of this size are amenable to all the host of tools we have available from dynamical systems theory. Can readily analyze their equilibria, the stability of those equilibria, and do they undergo bifurcations. So now, for the EMT network, what we have this master regulator Z, which drives the EMT. So it's mutually inhibiting microRNA MER200, and basically in connection in a larger network with snail and this other microRNA, you can define a set of EMT states that include the kind of ZEB low state, that's epithelial cells, and intermediate, and then the ZEB high state, in which cells undergo EMT. Usually these multi-stable regions define the regions of parameter space where you can have a distribution of cells, stable cells in more than one state, including the intermediate. The signal here is often TGF-beta in vivo and in experiments, in culture, but there's other growth factors that also induce EMT. Okay, so far I've told you we're going to work with simple networks, and we've got a great analysis techniques, right? So everything likely sounds really well constrained, but of course there's always a catch. So to hark back to the nice talk this morning from Russ, now we have to draw the rest of the owl. This is the hard part of model construction, and it's hard because it's subjective, right? There's not a principled way to do this, and this is combinatorics. Okay, here I'm showing you simple network. B receives signals from 3 things: activating from S, inhibiting from A, and it's got self-activation. Okay, so 2 ways to write that down. You've got dB by dt. You could combine these 3 signals by multiplying them. Multiplicative, okay, that's called AND logic. Or you could combine these by just adding them up. Okay, that's additive logic, additive regulation or logic. Okay, you've got this AND versus OR choice. And the crucial thing is I'm showing you here, I'm showing you the bifurcation landscape. The blue curve is the baseline and it's identical. Everything starts off the same. But when you perturb, right, we always care, there's always a perturbation. Then the red curves in each case, whether you choose the AND versus the OR logic, they could give you completely different self-fate distributions. This is a toy example I'm showing you, but the perturbed one is tristable for the AND logic and monostable for the OR logic, right, as you can see based on where that line lies. Okay, and we care because models of EMT in the literature of these networks use both. Okay, so how are we supposed to choose? Well, if we want to do things from a principled standpoint, then which logic should we choose? So the first thing we thought was, can we measure it? Maybe the answer is no, and let's go and look. Maybe there's literature on, do signaling factors combine additively or multiplicatively. We went and found there's a really nice paper from Arjun Raj's lab where they did this really careful experiment. They did RNA-seq plus attack-seq, where they just did that exactly this experiment: add two signals, TGF-beta and retinoic acid at different concentrations, and ask, do you see like the additive response or the multiplicative response, or something in between, or above and below, like sub-additive or super multiplicative? And the answer was yes, you see both. They saw these really nice peaks. So they saw targets of these two factors that are co-regulated that were additive, targets that were multiplicative, and they saw other things that were like sub-additive or super multiplicative, not very commonly. So you see these really strong two peaks, but with hundreds of genes responding in each way, so it tells you that, okay, well, yes, that this is how gene regulation is happening, but unfortunately, without going in and measuring exactly what your targets are and to what combination of signals, then we're not going to be able to, use the data to inform which is the correct logic. So now we want to study EMT. We want to study specifically the existence of these particularly metastatic intermediate states, and we want to understand the impact of logic on these networks. There's a simple EMT network that I'm showing you here. It's consisting of this mutual inhibition between A and B, which represent microRNA 200 and Z, with regulation from a signal S. And the key is here that there's many different paths. You can look at these bifurcation landscapes, as I talked you through, right? B represents like the EMT inducer, low state is epithelial, intermediate state and high state is mesenchymal. And we're really interested in the multi-stable properties, like the different paths of transition, which go through the intermediate state, the blue intermediate state, this is marking the tristable region, and which don't. So to study that, let's construct models. These are the AND versus OR logic models of this network, where basically, everything is kept as simple as possible, and these are just written down with Hill-type kinetics for the two different conditions. Okay, so when you do this, and then, as I said, you perturb the network, perturb one of these regulations on the network from the signal S from TGF-beta. First, on the left column, I'm showing you perturbing the direct activation for AND and OR. And basically, you see that for AND, it's the mesenchymal, the high state that changes dramatically. And for OR, it's the opposite. It's the epithelial state that changes dramatically. But then when you look to the second column and you perturb the indirect activation parameter, you get the complete converse, the complete different response, where now the mesenchymal state changes very minorly, and for AND, but it changes majorly for OR. Okay, so there's quite a lot of data condensed into this space, but the key point is how these bifurcations move controls EMT. It controls when EMT is initiated and the stability of the relative states. And again, the stability of the intermediate and the mesenchymal states define the metastatic potential. So on this slide, it's simply showing you that if you just, based on how you construct your model, with exactly the same model but a different way of combining signals, you can make completely kind of opposite inferences on the relative importance of the states and how EMT will progress. So basically, you better care about the logic in this cell state transition. So this is not just true for the EMT network, and this is in a classic repressolator model, where in a paper from the researchers a few years back, they showed that the requirement here is that you have multiple transcription factors expressed at a non-zero level. This is in neurocrest differentiation. And basically, that's for AND logic in their network, can you see concurrent expression of these multiple transcription factors? So again, this is where logic dictates the results of the cell fate in a gene regulatory network. And finally, we also looked at early embryonic fate decisions. And this gets interesting. So I've told you only so far about kind of pure logics, AND versus OR, but of course you can have mixed logic, which is a combination for more complicated models. And so previously in the Bessenard et al., they said that only this model on the left, logic one, is consistent with the data This is a bipotent cell fate choice in the embryo between primitive endoderm and epiblast, the blue versus the red fates. And basically, what we showed is that it's not the case that only one model is consistent. We looked at an alternative construction, okay, which is kind of subtle. It's up here. It's how do you combine these multiple signals regulating nanog? And in this alternative, for like the same parameters, the same conditions, you can actually get a tristable landscape, which is also consistent with the wild type data. And crucially, they have a lot of different mutants that they're able to compare with in these embryos. And of course, to test the model to predict, you ask, is it consistent with different sort of knockouts? And previously, they said the logic 1 was consistent, especially for the nanog knockout of these cells. That's the nanog minus cells, where you then add a receptor tyrosine kinase inhibitor. And it's true that if you add the receptor tyrosine kinase inhibitor to the nanog cells right here, it looks like at t equals 3, it looks like indeed you don't get a blue on state, which you should to match the data. But the catch is that we don't know what this, like, the temporal window is. So if we just extend the temporal window, like from 3 to 10 seconds, then you do get, even for this alternative model, alternative logic, something which matches the state. Okay, so I've just kind of hammered home the point that logic matters and how we consider cell state transitions and thus model plasticity, in this case, in the context of EMT. So let's go back to EMT and look again at metastasis and how we can actually identify what's going on through experimental design in live cells. So this data, I forgot to be the reference, this is from a paper from Bob Weinberg's lab, where they looked at stable clones derived from single cells that were either non-convertible, which meant they stay in an epithelial state, or they're convertible, which means they undergo EMT. Then basically we use these data to fit our model just to ask, at S1 through S8 are different values of different strengths of signals, so different concentrations of TGF-beta. And we can look for stochastic simulations, where does the first transition, like when do non-convertible cells become convertible? When do cells transition out of an epithelial state first? And basically, if we now look at the different logics, and versus OR, we can just vary the strength of the input signal, TGF-beta, and we can see that for OR logic, it's always the case that if you perturb the indirect interaction, you get a transition later. Okay, so Sx is greater than Sy, whereas for the AND logic, it's the inequality is reversed. It's always the case that when you perturb the indirect interaction, then you get transitions earlier. Okay, so we now have a way, we don't have to parameterize, we don't have to have any parameters in absolute units. We can just kind of do an experiment by perturbing, overexpressing ZEB or inhibiting a microRNA, and ask, as we titrate different levels of TGF-beta, which transition occurs first. And by this means, we can distinguish between multiplicative and additive logic in live cells. Okay, so to summarize, how can we use models to stop this nasty cell from metastasizing? I think we ought to develop simple gene regulatory models that are sufficient to explain the cell state dynamics present within a tumor. We have to fit models very carefully to data to make the best assumptions we can about logic. This is the kind of central point of this talk, as this will absolutely change the cell fates and the cell fate distributions that you have. Right, if there is a moral, it's this: it's that cancer will always co-opt all the sources of heterogeneity in the tumor, from epigenetic to plasticity to larger scale. And so I guess my last piece of advice is to start simple, but of course, I focused on one level of heterogeneity here, but of course the kind of ultimate goal is to grow larger models that start to incorporate, not only cell state plasticity, but cancer evolution and the wider context of the tumor evolving and transitioning between states in complex, noisy tumor on George Bloch's quotes, I just want to share my favorite, personal favorite, which is not about wrongness, but he did say that statisticians, like artists, have the bad habit of falling in love with their models. With that, I'll stop. And thank you everyone who contributed to starting.
Hi there. Thank you very much for the talk. It's always cool to see models with hysteresis and multi-stability. Would you mind going back to the slide where there was perturbations and horizontal shifting of the curve? Yes, this one. Would you mind elaborating a little bit more on this? I didn't fully understand the first time around.
Yeah, absolutely. So basically what we're doing here is like if in each row there's a baseline bifurcation, which is the black curve. And then for the two different logics, which are the two rows, we're perturbing one parameter by different amounts. So either the direct activation of Zeb, that's the string right here, or the indirect, which is here. And as you do that, you get like completely kind of opposing effects in how the bifurcations change in how the saddle points transition. So telling you like the stability, whether based on your conditional on your logic, you get completely different pictures of the stability landscape that you will miss if you only look at the baseline, but that will become apparent as you perturb the strengths of regulations in the network.
I think I see. Thank you.
Any other questions? Okay, well, let's thank Adam again. Okay.